our today's small seminar. Actually, this is a part of an activity that we have decided under a small grant supported by Baltic German University License Office between EMU Estonia and Claudia's Institute, the Federal Research Institute for Animal Health in Marense in Germany. So this will be kind of a small seminar which we have like 4 speakers speaking about different aspects of bovine and equine reproduction. And what the mode of today's seminar is actually we will have the 4 presentations, and then at the end we are going to have a kind of a moderated discussions so that the participants can either ask the question by themselves, or I'm very happy to even host the question through the chat box as well. So either way, you can communicate with us if you have any questions or queries. Right. Let me invite the first speaker today without any dues. So I think Andres is ready to do the first presentation today. Dr. Andres Gambini is a senior lecturer attached to the School of Agriculture and Food Sustainability. And of course, he has a joint position at the School of Veterinary Sciences, University of Queensland, Australia. So Dr. Gambini graduated as a veterinarian from University of Rio Cuarto, Argentina, and then completed his PhD in University of Buenos Aires, Argentina in 2013. Actually, then he moved to USA as a postdoc at National Institute of Environmental Health Sciences. And he worked there, I think, for 2 years, if I'm correct. And then he moved to Australia as a laboratory manager, dealing with, again, in vitro embryo production and animal cloning at the university in Melbourne. And then again, he moved back to Argentina, if I'm correct. Please correct me if I'm referring to wrong information, as a researcher in CONICET, Argentina. And then again, he moved back to Australia as a senior lecturer, where now still he's working at the University of Queensland. And moreover, he had been working as a scientific director at Onehealth, which is a company dealing with horse-assisted reproduction from Spain. So I know he has a lot of experience in equine reproduction. Actually, I was fortunate to see his seminal work a long time back in US, where they did the cloning of horse, I think from embryo splitting, if my memory is correct again. And then today, again, he's going to talk to us about equine reproduction under the title Updates on ICSI in Equids. Dr. Andres, the floor is yours.
All right, thank you so much for that introduction, Suranga. It's a pleasure to reconnect. It's been almost 10 years since we met in the States in the Frontiers in Reproduction course. So I'm really happy that after so many years, we have this opportunity to reconnect and share our research, and also to be in this session with another great speakers. So as Suranga says, I've been working with equine assisted reproductive technologies for some time now. And today's talk is going to be pretty much try to update where are we today on ICSI in horses, and I'm going to show you a few slides on what we are doing in other equids that are not horses and that are zebras and donkeys, right? So I think I have 20 minutes. Is that correct? Okay, so around 9:30, I'm gonna try to finish. If not, Suranga, let me know if right now time, right? Just to put all the audience in the same page, there are different assisted reproductive technologies. I've been working mostly with the production of embryos in the lab. These are one of these assisted reproductive technologies, and we have basically 3 ways of producing embryos in the lab. One of these ways is just mixing the gametes, the sperm, the eggs, IVF. We're pretty much all familiar with this technology. this has been had a huge impact in human fertility, and since 1978, more than it's estimated that more than 12 million people have been born by either IVF or ICSI, which is another of the technologies that I'm going to show you today. So producing embryos in the lab is becoming a very important thing in humans, but also in animals. And in the cattle breeding industry, IVF has been a game changer. We are producing more embryos in the lab than embryos that we are collecting by just traditional flushes on donor cows. And in horses, which is more our area today, IVF has been always a challenge. And recently, and I'm talking about 2 years ago, the group of Catherine Hinrich in the United States was able to elucidate some new protocols that are now able to capacitate the sperm, and this procedure is becoming more reliable in horses. And I know that in Belgium also they have been able to reproduce this process as well. But today I'm going to talk about actually ICSI, which it stands for intracytoplasmic sperm injection. This technology will just grab one sperm, inject it into the egg. And again, as you can see in my slide here, we have the human, which is, again, this technology is pretty much being used a lot, and horses, right? And among domestic species, horses is where ICSI right now is commercial, and it's growing a lot, and I'm going to show you the data on that. But and just because this has been also my topic for many, many years, and I'm still involved in this technology, just to show you that we can also make embryos in the lab now without the sperm, but by means of somatic cell nuclear transfer, and this is cloning. And again, horses here have a very important role because among domestic species, horses and dogs are the two main species where cloning is having a commercial impact, right? But why we are making embryos in the lab? So there are many reasons. I'm not going to spend too much time on this, but we can multiply the genetics of athletes. In case of horses, actually these technologies arise as treatment of infertility for mares, for stallions. This is very interesting, and I'm going to show you some data that we can, of course, try to understand more about assisted reproductive technologies, which are actually quite new, by using animal models and try to move on from the mice to larger animal models. It's also an excellent tool that is becoming more and more important in conservation of endangered species. We can use this technology to produce and do gene editing and produce animals that are genetically edited, and of course for genetic preservation, right? So all these together are pretty much the reason why this technology have been growing a lot, and particularly in the equine breeding industry. And this slide shows us how ICSI looked like before 2010. So we're talking about 13 years ago, right? So as you can see, a few labs around the world were able to handle this technology, and they reported the birth of foals through ICSI. And here we have the first one that was produced in the United States, and then a few other countries, including Australia, Italy, and Belgium. They were able to produce horses through ICSI. But this is how ICSI looks like right now. So just 10 years after, pretty much we have foals being reported in a lot of more countries, almost in all continents. And of course, there's a lot of information that can be, missing in this slide. It's becoming harder and harder to really get the right data, and we are working on trying to do something about this and try to collect more precise data. But what you can see in brackets is the number of laboratories that are, as far as I know, aware that exist in each of these countries. And also in brackets in some countries are the number of embryos that were produced just on this year, in 2023. So as you can see, there are a few countries that are the bigger producers of embryos through ICSI in horses, but many other countries are starting to set up labs and produce embryos. The IATS, it's one of the societies that is responsible for collecting data about this, and it's sometimes the numbers, they don't look real, or it's very hard to collect, and or collecting data that it's to reflect what is really going on in the industry is actually quite hard. But this is the data from 2022, so it's reflecting the number of 2021. In horses, these are the numbers of embryos that are produced in vivo with traditional embryo transfer, and this is in vitro production of embryo. And just to show you that this in vitro production is growing a lot. And I recently gave a talk in Chile, we had the World Equine Veterinary Association Congress, and I was collecting data for that meeting, and I'm gonna share that data today to you guys. Again, all these numbers are estimates because it's really hard to get the right ones, but there are probably about 33 laboratories now worldwide, and probably we are producing more. We have produced more than 50,000 embryos so far through this technology, and the demand keeps increasing. So I'm going to take you through very quickly through this process, just to understand why ICSI is growing so much, and what are the advantages and the disadvantages of this technology. So basically, because we are producing embryos by fertilization, we need sperm and we need oocytes. We know that we can collect sperm, right? And we can preserve this sperm, although we still have some challenges in horses. Pretty much this technology has been settled. And the good thing about ICSI is that we can maximize the use of sperm, and we can make good use of semen collected from the epididymis. But also we need eggs, and this has been a little bit more challenging always in horses. But now again, this technology has it's more established, and we can do this by means of OPU, pretty similar of what is being done in the cattle, with a few differences that I'm gonna try to take you today. And something that Danielle will talk about today is the possibility or not, possibly in the future, of oocyte preservation. We know that we can preserve the sperm and have frozen sperm, but oocyte cryopreservation has been more challenging, and this definitely gonna change even more the horse breeding industry if we can make it commercially successful. So then what we're gonna do is gonna grab one of the sperm, we're gonna inject it into the mature egg. We're gonna have an embryo. That embryo is gonna grow in the lab for 7 to 9 days. We used to do, back in the days, early embryo transfer in oviducts. We are not doing that anymore because we got pretty efficient on growing embryos in the lab. So we just pretty much cryopreserving in vitro produced blastocysts, which are very resistant to cryopreservation, so they can go through this process very well, and then we can just do embryo transfer, as we all know. So basically, understanding the technology, we can if we can get oocytes from follicles in the ovaries, pretty much we can use and we can produce embryos if our mare have follicles in the ovaries, and that's kind of the idea, although it might be a little bit more challenging than this. But if our mare has follicles in the ovaries, we can potentially produce embryos out of her. So we are bypassing a lot of the potential problems that we have in vivo, in the oviducts, in the uterus, and so on. And this technology, as I said, initially was, targeting all mares with fertility issues, but this is not necessarily the main reason that we are using ICSI right now in horses. So right now, we are collecting oocytes from young mares and producing embryos from young mares when we know that these mares are fertile, because basically the idea now is to preserve genetic, right? And a lot of what's going on right now is to have a genetic bank. A lot of horse owners are producing embryos, and they are just starting to store embryos in liquid nitrogen tanks to sell in the future. And I don't want to spend too much time on this, but one of the great advantages of ICSI is that we are starting to have a non-reproductive season in horses, or we can maximize the breeding season by other more traditional assisted reproductive technologies like embryo transfer, artificial insemination, and so on. And then during the transitional periods, as you know, mares are seasonal breeders, and we have transitional periods where mares are not really cycling, but we have a lot of follicles in the ovaries, and we can maximize these periods that normally we can produce embryos, and now we actually can with ICSI. But also ICSI, and this is how ICSI arises in humans, right? ICSI is actually an amazing technology for the male, because in ICSI we only need one sperm, so ICSI can help us to generate offspring of stallions that have really, really, really low quality semen. But again, as long as we have sperm, and that sperm has proper DNA, we can make embryos. And this has been one of the reasons of why ICSI has grown a lot in horses as well. Why? Because we can maximize the use of semen. There are different ways that we can do this. I don't have much time to get into the details, but of course, we don't need a whole straw anymore. We can just cut the straw. We can throw one straw, redo that straw, and make many other straws from just one. And this has been very important in the breeding industry, because now we can produce many foals out of just a single straw. And there are a lot of very good stallions with an high valuable genetic that these stallions are not alive anymore. And the only thing that we have from them is semen in liquid nitrogen tanks. So we now can produce a lot of foals from these stallions that otherwise we will, be spending to produce just one offspring. And another big thing that it's coming, and it's becoming, and I think that in the next few years, we're going to see this increase a lot, because the sperm sorting technology is getting better, and it's not damaging the sperm as much as it used to do, is the use of sex semen to produce foals. Sex semen has been always not quite efficient in horses, but the possibility of using that semen in the lab for ICSI, it's becoming a huge thing. And then there are, more novel or different ideas that we can use the semen, that we can produce embryos in the lab with semen that it's actually dead. And we really need to kill the sperm before injecting the sperm into the egg. So we don't need live sperm. And there are different and novel methods of sperm preservation, like freeze-dried sperm, that has been used in many species, including the horse. And it's possible to produce live offspring out of this. All right, so how we collect oocytes, I think that all of you are pretty familiar with ovum pickup, right? And transvaginal oocyte aspiration. This is the technology, and the concept haven't really changed too much, But what had happened is that we got much efficient, and I think that this helped us the technology to increase as well, and to be more commercially available and more not too expensive, I would say. And it's also improving in a sense that we are reducing the risk for the mares, because we are able now to do this technique faster than we used to do. So I think that from animal ethic concerns as well, this is getting better and better. But basically, we can collect eggs in our mares from every 2-3 weeks. We can get an average of 10 oocytes per mare if we have some criteria for selection before OPU. We, of course, have to be aware that this is a procedure that we are inserting a needle inside of the abdomen of the mare, right, to get access to those ovaries. So it has risk. This risk can be very minimal if we do the things right, but it's something that we still need to advise to the owners that that their mare is going to be subject to a procedure that has risk. And we are using actually a quite big needle that we're gonna get through the vagina into the follicles. We're gonna aspirate, and the challenge here has been always that oocyte, it's very attached to the follicular wall in the ovaries in the mare. So it's very hard just to get oocytes by simply aspiration of the follicles. So we need to flush the follicles 5-8 times. We need to scrub the follicular wall. So compared to the cattle, some of you might be more familiar with cattle, this process takes longer, and follicles in the mare are bigger, so there's a big volume. It's a little bit more complex, but we are getting this technology more set up now. Now we'll have to hurry up. All right. So one of the things that have helped a lot for this technology to spread out and how many labs start working is that we can, once that we collect the oocytes from the mare, we can just put these oocytes into a vial like this one here in just flushing media or embryo holding media, and we can keep these oocytes at room temperature and ship these oocytes to the lab, and they will be fine and happy for 24 hours for sure. We are now seeing some new research that they are trying to extend this time. It's still a little bit controversial if they can, survive longer than 24 hours. But if you think about it, in 24 hours, you can cross many countries in some places. It's very important to keep the right temperature. But again, Daniel will give us a talk today, and this will be very important to have it commercially available soon. After the oocytes arrive to the lab, we need to do in vitro maturation of these oocytes. What you're seeing here are oocytes that are maturing in the lab. You can see how the cumulus cells are expanding. That's what we want. We are simulating what is going on in the pre-ovulatory follicle. And basically, we did some studies where we show that about 30 hours of in vitro maturation is sufficient to have most of our oocytes with the first polar body extruded. So that's pretty much, I guess, the time that most of the laboratories are in vitro maturing oocytes right now. So once that we have a mature oocytes, and we know because our oocyte will have the first polar body, we're going to inject the sperm. There are many ways of doing ICSI. A lot of the laboratories, they will use a piezo drill. So they can use just a plant pipette, and I've been doing ICSI most of the time, just as conventionally being done in humans. You just need to be sure to break the membrane of the egg, so the sperm is being injected inside the oocyte, inside the cytoplasm, and not outside. And that sometimes it's not something easy to be sure of it. Something that we are asked a lot is how much the whole process takes. And because when you do OPU, and the owner will take, okay, when I will know if this technology is successful. And you have to tell them that you need to be patient, because pretty much you have to wait, for sure, in most of the cases, 9 days, because all the times that I've been explaining a little bit. So we have the holding medium, the holding time, the 30 hours of in vitro maturation. ICSI itself is very fast, but then we have to grow the embryos in the lab for 7 to 10 days. And vitrification also is very fast. But if you take all this together, at least 9 days is the day of OPU. And this slide here is showing us what we can expect if we have a good setup, right? Not all the groups can achieve this. These are reports for groups that have a lot of experience in OPU and ICSI, but it's a good reference if you are starting to do this technology or planning to set up an ICSI lab and team up with people that it's doing OPU. And you have here a reference of the efficient of each of the steps of this technology. But I want to focus here a little bit. This is important. So 25% of the mares that you do OPU, they will not give you an embryo. And you have to say this to the owner, right? So you want to advise the owner that maybe you're doing OPU and that mare has a 20% chance of not having any embryo, no single embryo, right? But because the rest, the 70% of the rest of the mare they do will give us embryos, not only one, but most of them will probably give us more than one embryo. This technology can potentially give us 1.5 up to 2, and maybe some labs are getting even higher embryos per OPU session. And this is getting more efficient and more cheaper than conventional embryo transfer in the horse. Just remember, if you're more for a bovine reproductive biologist, that in the horse, we don't do super stimulation of our mares, so we only collect one embryo per cycle. So having the opportunity to have 2 embryos per OPU, it's very competitive with this other technology. All right, so Suranga, if you allow me 5 minutes to just show a little bit of the data that we are doing.
Please go ahead. Please go ahead. You can.
Yeah. Okay. So for those that are not aware, equids, also horses. In the equid family, we have horses, donkeys, and zebras. And we have 2 species that are domestic, horses and donkeys, but we also have wildlife species. We have zebras, they are all non-domestic species, different species of zebra, 3 species. And we also have wild species of donkeys, African donkeys, Asiatic donkeys. And particularly the gravy zebra and the African wild ass are critically endangered. So there are an estimate that less than 200 mature individuals of African wild ass and maybe 2,000 gravy zebra left. So because we have been working with horses for so many times, we decided that, okay, we can maybe apply assisted reproductive technology to try to preserve this species. And partnering up with a zoo here in Buenos Aires, as a male zebra died, and as you know, we can collect sperm from the epididymis, and we can grow cells from the skin. And what we did zebroids in the lab. So we injected sperm from zebra into horse eggs, and we did zebra cloning by injecting a zebra cell into an enucleated horse oocyte. All these results were published, and I can share you guys the manuscript, but we were very successful on producing blastocysts by either ICSI or cloning in zebras. What we are doing more recently is collecting oocytes in donkeys. Doing OPU in donkeys is a little bit more challenging than doing OPU in mares. We need to have some considerations about this, but we can use the domestic donkey as a model to try to understand reproductive physiology and in vitro production for future applying this in wild donkeys. So I'm going to skip some of these things because I don't have much time, but we are studying differences between donkeys, young mares and donkeys, and between young donkeys and old donkeys, to try to have this idea of using the donkey as a model for human reproduction. Donkeys have a longer span life than mares, and they can be used as a potential model for understanding aging, for example. And here we are doing some experiments where we are looking at mitochondrial activity and the potential membrane of mitochondria of horses and young and old jennies. And I'm just gonna quickly share some of the results. But what we are seeing is that even though old donkeys have more total mitochondrial, they pretty much, the healthy mitochondria, the one that are active and healthy, are pretty much the same. So at the end of the day, the number of active health mitochondria that our old donkeys have is lower compared to young donkeys. And we are trying to understand if this is going to have an impact on in vitro embryo production in this species. And for that, we needed to set up ICSI in donkeys because there was no report until this year of blastocysts produced by ICSI in donkeys. And we were successful on producing the first donkey blastocyst in vitro. Not really big changes here, it's just that the donkey oocyte takes more time for in vitro maturation. And we are trying to use whatever we are using in horses for donkeys. Probably we need to, understand a little bit more what the donkey needs, that it's different compared to the horse. And we had the opportunity to do some time-lapse imaging and to see when the embryos are actually achieving the different embryo stages. This data is all published and you can have access to the manuscript. And finally, I'm going to show you that we are also doing hybrids. That a fascinating thing about the equine family is that they can all intercross. You can mix horses with zebra, zebra with donkeys, donkey with horses. And in this case, we reported the production of the first mules in the lab. So we injected donkey sperm into mare oocytes, and we didn't see any difference in development. And yeah, sorry that I'm rushing out, but I just want to keep it on time. Just because now I'm in Australia, I just want to show you for fun that ICSI is also an amazing tool for research in general. So when I arrived to Australia a year ago, I realized that there was sperm in liquid nitrogen tank from koalas, and I decided I need to, inject this sperm into and start setting up ICSI in this species. So just for you to see that a koala sperm there, then I wasn't even sure that the koala will enter into the ICSI needle. Anyways, just to conclude, I show you how ICSI is increasing in the horse breeding industry. It's becoming a really important thing. Of course, you have some breeds that they will not allow the use of assisted reproductive technology, but in the breeds that they are open to this technology, this is growing, and we can use this technology and everything that we are learning in the course to apply this in other species like zebras, donkeys, mules, and so on. And just, I don't have much time, but there are a lot of things that we don't know and that's the reason why we still need to do research on this. All right, thank you so much. Apologize for getting over the time. It's usually me. But yeah, I guess questions gonna be in the end, right?
Yes. Thank you very much, Andres. It was a very fascinating story. So I'm sure our audience will have some more questions later. So now we'll move to the second speaker. Our second speaker is Professor Dr. Claudia Klein, who is heading the Institute of Farm Animal Genetics in Federal Research Institute for Animal Health in Mainz, Germany. So Dr. Klein graduated with honors from JLU Gießen, Germany in 2004. And then she got her thesis, thesis entitled Identification of Genes Induced by the Concepts in the Bovine Endometrium during the pre-implantation period from, again, LMU Munich. And then after that, she worked as a clinician in the Clinic for Obstetrics and Gynecology and Andrology of large and small animals at the same university. And afterwards, she joined with the University of Florida for her residency in thyrogenology, where she obtained her Diplomate of American College of Thyrogenologists in 2009, and also at the same time from the European College of Animal Reproduction. In 2010, she defended her PhD in the US, and then she worked as a postdoctoral associate at the University of Kentucky. And then afterwards, she moved into Canada to join the University of Calgary at the Faculty of Veterinary Medicine, where she promoted up to associate professor before she moved into Germany, and now she's heading the Institute of Farm Animal Genetics in Mariens, Germany. So Dr. Claudia today will be talking to us about single-cell sequencing of bovine embryos. Dr. Klein, the floor is yours.
Thank you, Saranga. I guess we sort of come a little bit at the later end after having done ICSI. We're interested in the impact of the assisted reproductive technologies on embryo development. And I guess that's not really a new question or a new topic, but we have a plethora of new skill sets available at the molecular biology level to assess the effects of assisted reproductive techniques on the embryo itself. And so one of those really fascinating techniques is single-cell sequencing. And due to a lack of forces at our institute at the moment, we're using cattle as a research model. And just for those of you that aren't familiar with single-cell sequencing, I'll give you a brief introduction. It's a really clever technique, and the way it works is that you need a single-cell suspension and a microfluidics device. So on the one side of the microfluidics device, you feed the single-cell suspension that you've generated, for example, through enzymatic digestion of an embryo. And on the other side, you're feeding so-called gel beads that the 10X technology utilizes. And then, and the third sort of a pipeline is oil is introduced. And so now you're forming an emulsion. So within the oil, you have an emulsion where you have these little micelles that contain one of these barcoded gel beads and hopefully a single cell, because you've created a single cell suspension. And these barcoded gel beads, they are sort of the key. These beads are covered with hundreds of thousands of little capture nucleotides that contain a barcode, and the barcode is like a house number. So each one of these tens of thousands of barcodes has a different identity. So it's almost, yeah, it's like a house number. And then there is a poly-DT tail for capturing the mRNA. And so the next step, once you've created this, suspension, you have thousands of reactions happening in parallel, where you lyse the cell, the mRNA is released, it's captured by the poly-DT tail on these beads, and you create the cDNA. And then the way it works is that the cDNA is then sequenced, and when you do the data analysis, due to each cell having an individual barcode assigned to it, you can then assign, okay, the transcripts with the barcode, 12 belong to this cell. The next cell has barcode 13 and so forth. So you can assign the transcripts to the cell of origin where they come from. And so our sort of general interest is that we revisit at the genetics, epigenetics, and proteomic level the impact of the assisted reproductive technologies on embryo development. Again, it's not a new topic, but we have now a plethora of new tools available to examine this question. And again, we're using here the cow as a model for human, partially because this is our biggest funding agency in Germany that we can get money from. So of course, the research itself is also of interest for the bovine industry, which in Germany is not quite as developed as, for example, in South America, but it is increasingly being used in Germany as well to multiply genetically superior animals. Initially, we had planned to run the analysis on using blastocysts, but that sort of miserably failed because it's just really difficult to create a single cell suspension from blastocysts. The cells are like little magnets, so as soon as you've trypsinized them or whatever, they tend to stick back together. And key to this technology is that you have a single cell suspension. So we moved on to using day 17 elongated embryos, which also sort of addresses the interesting question, once you've generated a blastocyst and you've transferred it to the recipient uterus in this 1 week phase, maybe some of the changes that we have introduced through using ARTS are also being reversed. For those of you that haven't seen an elongated cattle embryo, at day 17, they are of impressive length. And you have a hard time finding this embryonic disc. You can see it here. And so for this analysis, we dissected out the embryonic disc and separated and digested it separately from the trophectoderm in order to have an adequate representation of both cell types. You can feed about 10,000 cells per sample into the system. So we wanted to make sure we have an adequate representation of the inner cell mass and the trophectoderm. And for now, I'm just showing you data from in vivo derived embryos. So these were cows going normal artificial insemination, and then the cows were sent to slaughter at day 17. We retrieved the embryos and we examined 3 biological replicates to first define what the normal transcriptome looks like at the single cell level. So for those of you that are maybe a little bit bioinformatically inclined, 10x Genomic comes with a very good analysis pipeline, and there's sort of a standard package from the Surat laboratory that has a lot of algorithms written. And so, of course, sort of the first step is alignment to the genome. And then we noticed that when we were working with bovine embryos, we had to apply an algorithm called SoupX, because even though we had a single cell suspension with cell viability over 90%, some of the trophectoderm cells got damaged, and they're so packed with transcripts, especially interferon tau, that these are leaked into your suspension. They can stick to the outside of the cells, so you have sort of a contamination. But there's an algorithm available, SoupX, that can sort of remove contamination. And then we ran sort of some quality parameters. For example, we only included cells that contained less than 10% mitochondrial genes, because by this means you can exclude cells that were damaged. It's the way it works is if you have a damaged cell, the membrane is leaky, so you're losing your cytoplasmic transcripts, but the mitochondria is still trapped within the cell. So when you sequence a dead cell, you have a really high percentage of mitochondrial genes. And this is a means in which you can filter out dead cells, because no matter how gentle your digestion is, you will always have a few dead cells within your suspension that you want to remove from your analysis further down. And that is really the strength of the single cell level. You can really pick individual cells or a bunch of cells and remove them from the analysis when you, for example, identify that they are damaged cells. 10x Genomic comes with a very good interface that you can also analyze your data when you're not a pro in R. It's called the Loop browser. And so this is just an example of sort of what our initial data looked like. You can then type in marker genes that you know, for example, is only expressed in the trophectoderm, like interferon tau, and then it highlights the expression of interferon tau within cells. And so you can easily see, for example, this cloud here is obviously trophectoderm. Here we have OCT4 as an inner cell marker. Here we have vimentin as a mesoderm. So you can see our approach that we did a separate digestion from the embryonic disc, and the trophectoderm was successful because we find all of the different germ layers, including the trophectoderm, reflected in our data set. And so, for example, the way this works now, we had 3 biological replicates. And so you can see here, I didn't circle any marker genes, but the cells within the circle are the embryonic disc. And then to the right-hand side, this is the trophectoderm. So you can see just by bare visualization, the embryonic disc sort of a UMAP projection is very similar. Within the trophectoderm, we have a little bit more heterogeneity, which probably is reflected at day 17. We had really over 1 million cells in our trophectoderm cell suspension, but we're taking less than 10,000 out. Sort of reflects that there's some heterogeneity present in these cell types. When you're working with multiple embryos, you run a so-called integration data workflow. And so it pretty much overlaid the data of all our 3 embryos, because we were not interested in differential gene expression between our 3 in vivo embryos, but we rather wanted to define like what are the different cell types that we can find at day 17. And again, then sort of you can play to your heart's content. Again, you can see again on the left-hand side is embryonic disc, on the right-hand side is trophectoderm. So you can see sort of a big cloud of what we call core trophectoderm cells that are interferon tau positive. And this cluster down here that's splitting off, these are binucleate cells. So you can see the classic interferon tau positive cells are also positive for PAG12 as a pregnancy-associated glycoprotein marker for mononuclear trophoblast cells. And then in the cow, we have the particular occurrence of these binucleate cells. PAG18 is one marker. So you can see at day 17 down here, we have the binucleate cells already showing up. You can then really go into depth and analyze, for example, individually. That's the beauty. Now we've split up our data set. We're taking the embryonic disc and analyze it separately from the trophectoderm. And again, here you can identify your different cell types based on known marker genes. Same here, OCT4 for inner cell mass, MES1 for the developing mesoderm, vimentin for the mature mesoderm, and up here in this corner marked by fibrinogen beta is the developing endoderm, for example, which you can find. We were initially surprised that we found very little endoderm contribution in our sample, but there is also a publication that endoderm development in cattle is remarkably slower compared to other species. Then again, you can even find hematopoietic precursor cells at this stage of development up here. These cells are also vimentin positive. They're of the mesoderm lineage, but they already express markers that are characteristic for the hematopoietic precursor cells. Because for our third-party funding applications, we're trying to use the model as the cow as a model for human. We integrated our bovine data with a published single-cell data set from a human embryo. This is a pretty cool human embryo. This was a pregnancy abortion that took time very early on. sometime between 16 and 19 days after fertilization, and they retrieve the embryo. And then they perform single cell sequencing. And this is, to me, still one of the most fascinating images that we've created so far. In gray are our bovine cells from the 3 in vivo embryos, and in the color are the embryos, are the human cells. And so you can see how they really overlap. And at this early stage, this algorithm that sees, okay, these cells are similar, they overlap, cannot really distinguish whether you're using cow data or whether you're using human data. So then the nice thing is, because the human cells are very extensively annotated with different marker genes, whether, you know, what kind of mesoderm it is and so forth, we can draw back the conclusion of our bovine cells. For example, you know, do they belong to axial mesoderm, nascent, emergent, advanced mesoderm, and so forth, which would have been very difficult to do without the human data set where we ran the integration. Again, to your heart's content, you can look for marker genes and identify them. So the embryonic disc is sort of the easy part of the embryo to evaluate. Currently, we are trying to figure out a new cell population in the trophectoderm that we've identified. Again, these are trophectoderm, the data from the 3 embryos merged. So we have the core trophectoderm here marked through interferon tau expression, so fairly simple. We have up here, this is what I highlighted here, so the developing binucleate cells branching off. And then up in this lower left corner is a cell type where we are certain these are trophoblast cells, because 2 of our embryos were male. We can sex the embryos based on the transcription. And these cells in that lower left corner are also male, so they're definitely trophoblast cells. So they're not a contamination with endometrial cells. They are GATA3 positive down here. You can see that. So they are of trophoblast origin, but they're not interferon tau positive. They're not positive for one of the pregnancy-associated glycoproteins. And interestingly, we also did a direct comparison to human trophectoderm cells. So these were single cell data that had been published where people took blastocysts, placed them in culture, and then created sort of trophectoderm outgrowth. And these human trophectoderm cells are staged to be roughly from 13-day-old embryos. And we did the same workflow here. So we did a data integration, and this is even more fascinating to me. You can see in gray are the bovine cells, and the colored ones are our bovine. The gray ones are the bovine, and the colored ones are the human cells that come with the annotation. And so you can see here in pink, the TE is like the classic trophectoderm, and then over more to the right is the specialized cell types that you can find in the human placenta. The CTB stands, for example, for cytotrophoblast, and the STB is the syncytiotrophoblast. And we can find our cell population where we don't know what it is. One of the marker genes for this cell population up here is WFDC2. So if we, after this integration workflow, take a look, where are the WFDC2 positive cells? You can see they're up here in this little gray corner, so the bovine cells, and they sort of align here with these primary fusion competent, respectively, those primary cytotrophoblast cells of the human placenta. And then encircled in red, just for your information, these are the binucleate cells. You can find them down here close to this migratory trophoblast cells that are present in the human placenta. So I guess for our external funding applications going forward, I think we can show these two images, and I think at least at the single cell transcriptome level, at the pre-implantation stage, I think the bovine is a fairly accurate model for human development. So right now we're analyzing in the next step the differential gene expression, where we're comparing in vivo with in vitro and so forth. Again, this is the unknown cell population. I mean, I'm almost having sleepless nights over that cell population, because we would really like to know, like, what is the function. And so in order to figure out where is the cell population coming from, down here is all 3 embryos integrated. Up here are the 3 individual embryos that we have. And because we didn't exactly time ovulation, like we didn't do an ultrasound like every few hours, the embryos that we collected can be sort of 12, maybe up to 20 hours, maybe even 24 hours apart in exact age. And that is reflected. We can see we have one embryo, the one to the left here. That's one of the very early, like a little bit younger. It also doesn't have any advanced mesoderm. And the embryo all the way on the other hand to the right is the most advanced embryo. And so you can see this big cloud here is the interferon tau positive. And you can see this cloud here is the developing binucleate. And you can see, especially for the young embryo, this unknown cell population seems to originate out of this binucleate cell population. But due to lack of time, I'm not showing all the data. Interestingly, even though they develop out of this binucleate cell population, based on pathway analysis, these cells have a stem cell character, whereas this developing binucleate cell population is our terminally differentiating cells, whereas again our unknown population has a stem cell character. So what we did right now is I'm picking up the samples on Monday. We collected sort of a day 40 embryo after slaughter, and we're doing in situ hybridization to see like where can we actually see them in situ when we have trophectoderm from a bovine conceptus. And that was it for today. Acknowledgements go to Doris Hermann for the technician who does the single cell sequencing, Sonia who did the IVF in the lab, and Gregor who is doing all the bioinformatics analysis, because that is way above my pay grade, so to say. It's lucky to have good PhD students that can run the analysis.
Thank you, Claudia. That was a very fascinating story again. I'm sure there must be some questions, but please keep them at the end of the session. And I'm again reminding you, you can either ask them by yourself, unmuting yourself for later, or you can even post them in the chat box as well. So the third speaker today in our seminar is Dr. Daniel Angel-Velez from Department of Reproduction, Obstetrics and Herd Health at the Faculty of Veterinary Medicine from the University of Ghent. Dr. Daniel obtained his first veterinary degree from Universidad CES from Colombia, and then completed a master degree in equine veterinary medicine at the same university. He then obtained his PhD from University of Ghent, Belgium, and currently he's working as a postdoctoral researcher at UGent as well. So he has been working on equine reproduction for a long time, so he has a lot of experience to share with us. And so today, Dr. Daniel will be speaking to us on introduction to oocyte cryopreservation. Dr. Daniel, the floor is yours, please.
Hello, thank you very much for this opportunity. It's a great pleasure to be here and to share a room with amazing speakers as Dr. Andres and Dr. Claudia. So today, let's see if I can share the screen. Yeah, this one.
Yeah, you can see it perfect.
Okay. So we have got fascinating conference. Now I'm trying today I want to speak about oocyte cryopreservation. In humans, this is a reality, so it's quite difficult to get recent data. But if you see in United States from 2010 to 2015, the number of cycles that are or IVF cycles that are performed with cryopreserved oocytes grow in a rate of 800%. So more or less in humans, they are almost taking the all freeze all policy. But unfortunately, this is not happening in domestic animals still. So I'm trying to give you some key factors in oocyte vitrification to try to work in any species. I have focused my work mostly in equine, but I'm gonna work in the key factors more or less in all the species. So in nature, especially in horses and cattle, we only have one oocyte release every cycle, and we only have a single chance of making a baby. And in mares, it's only during certain time in the year. So this limits a lot the genetic, the spread of the genetics and the genetic conservation if we want to do it. But why we are interested in conserve the genetic of these animals? Well, because if we go for genetic conservation in equine, for example, Andres gave us an amazing talk, and the ICSI has grown a lot because we can have champions mares in the Olympic Games, which a fall from them cost millions of euros. So that's the main reason of doing genetic conservation. But also we have breeds that are in danger of extinction, or some breeds like in cattle that are super amazing producers that we want to preserve and we want to multiply. So these are only some reasons to preserve the genetics. How can we preserve the genetics? Well, we can do it by the oocytes, either in immature stage or mature, even in vitro or in vivo maturation. We can preserve the male part with the sperm, and we can preserve the embryos if it's in vivo produced or in vitro produced. Today, I'm gonna focus in the female part because the male part, as Andres explained, there is quite, I mean, it's still room for improvement, but it's quite established, and there is millions of cells in one ejaculate, so you can preserve quite easily. But in the female part, we have the embryo, which obviously preserving the embryo gives a lot of advantage, because if you are talking about cows, you need fewer recipients. In endangered species, you can do embryo banking. In all species, you can just move embryos around the world instead of moving the standing animal, and it's much easier and safer. And you can avoid out of season offspring, which is very important, especially in some breeds. And obviously, as Andres said, pre-implantation genetic diagnosis, which is crucial, and it has been growing a lot, especially in cows, because you can select who is gonna be the good producer just before transferring. The point is that you require both parties. Sometimes you don't have the male, and it's a question because it's an advantage or a disadvantage is that you retain one progeny. So you don't know how it's going to be the genetics. Obviously, you have the genome that is going to predict in cattle, for example, if it's going to produce a lot of milk or not. But it still is a combination of genetics. Differently, when you have the oocytes, which is the only way to truly preserve 100% the female genetics. And here, you don't need to think in what male I'm gonna use. So this is the main reason for preserving oocytes. And a part of that, obviously, if you preserve oocytes, you are much more flexible, especially for example, in horses, in which you can move oocytes through the world, and we can have countries like Colombia, like Argentina, where you have much more availability of oocytes, and move it to here to Belgium, on to United States, or to Australia, where we have all the facilities to do ICSI and to do high technology research. Obviously, you also can give it the opportunity of a mare that just died, or save the last rhino, which is for conservation purpose. So there's obviously a lot of reasons, and then the question obviously is how we preserve the genetics. And this is done by cryopreservation, techniques that allow us to preserve cells over the time, keeping their viability. And there are several techniques, but basically, in oocytes, we have two kinds of techniques, slow freezing and vitrification. In the slow freezing, the main difference between them is the cooling rate. As you can see in the graph, the slow freezing, you make it in a program machine with a program cooling rate, in which extracellular ice formation is initiated in an organized way. However, this has a lot of risk, because there might be the ice formation, then the ice crystal can damage the cell. On the other hand, you have vitrification, which is a super fast, ultra rapid freezing, in which you transform liquid into glass. But for doing that, you need cryoprotectants, and high concentration cryoprotectants might be toxic. But we need to know which of those two techniques to use. And in humans, where oocyte cryopreservation is now a clinic technique, they have done research on it, and they have demonstrated that vitrification is associated with a higher pregnancy rate. More recent papers also have shown the difference, and basically, I could say that all the centers in human IVF, are using vitrification as a chosen technique for cryopreserved oocyte. But for doing a stable vitrification, which means no ice formation, you need to combine biological and technical factors. And that's what we are going to focus today. So for the biological factors, we talked about meiotic stage and presence or absence of cumulus cells. In the meiotic stage, it's clear that the best oocyte to vitrify is the in vivo mature oocyte. Is the one that they use in clinical practice in humans. It has been demonstrated in equine that is also the best. The first fall from vitrified oocytes was from an in vivo oocyte. Then later, the same group in 2010, and recently in a paper, they showed that they have comparable blastocyst rates and pregnancy rates from from vitrified oocytes. Also in mouse, in different papers, they have shown no difference in blastocyst rate and similar transcriptome after vitrification. But if this in vivo is a limited technology, especially in equine, in which you can only have one oocyte per cycle. So shall I choose GV or in vitro mature oocyte? This is a question that we have no address still. In cows, for example, it's very contradictory, the information. You have several papers that you find better blastocyst rate in mature oocytes, but others that you have better blastocyst rate from immature oocytes. And this is gonna depend, I think, in the protocol. This is gonna depend in the cryo device that you use, in the cryoprotectants that you use, quite different things. You have in pigs also contradictory information. Sometimes better in GB, sometimes better in mature oocytes. If we go to mouse, equine, and goat, it seems that there is a subtle advantage to vitrified in vitro mature oocytes. Most of the research, especially in equine, has been done in GV because it's the most practical in terms that you can vitrify it in field conditions. You don't have a laboratory to mature the oocyte. But a couple of work and a recent work that we did, we found better efficiency in vitrification of mature oocytes and a similar and more similar transcriptome in the mature oocyte. In goats, there is only one paper and they got better blastocyst rate in mature oocyte. And in mouse, there is less epigenetic change in GVOs. Also, there are less epigenetic change in GV oocytes. They have lower survival and blastocyst rate, and they have lower quality blastocysts when they came from vitrified oocytes at GV stage. This is the work that we did with transcriptome, and fortunately, we didn't find too much difference between immature oocyte and fresh, and mature oocyte and fresh. But as you see, we found less number of genes differential expressed in vitro mature oocytes versus fresh, which give you a subtle advantage of vitrifying in vitro mature oocytes. Then if we move to the cumulus cells, in humans, they didn't find difference in survival, but in mouse, cows, and sheep, they found a reduced fertility after IVF in the neutered oocytes, and same in cattle and sheep. So here there is a point for the method of fertilization. So if we go to equine, the first paper that evaluated this was the group of Utrecht, 2006, and they found a protective effect. So they found less damage in the spindle when the oocytes were cumulus and closed. But after that, I work with in my laboratory, we found that if you have a lot of cumulus cells, you have lower maturation rate. Then that's why most of the work with equine has been performed with partially denuded oocytes only with the corona radiata. And this is in equine. All the embryos that we have produced so far with vitrified oocytes are with corona radiata. But we perform ICSI after maturation, and we vitrify immature oocytes. In cows that we tried also with in vitro mature oocytes, we also had a control of fresh oocytes, but that we partially denuded to see what was the effect of that. And we found that partially denuded oocytes in kind of individual culture, which was the time-lapse dish, and in group culture, we have lower blastocyst rate in the partially denuded oocytes. So the cumulus cells the number of cumulus cells are important. If we move to the technical factors, cryoprotectants, cooling rate, and viscosity, we have to center first in cryoprotectant and show quickly how they work, because they are the main point of cryopreservation. So in cryopreservation, we have equilibration, vitrification, and warming, and 3 key factors: water, permeating, and non-permeating cryoprotectants. The permeating cryoprotectants, what they do is replace water and decrease the freezing point, exchanged by water and during equilibration and vitrification, and then has to go out during the warming. The non-permeating cryoprotectants, they help in the last step of vitrification to take out the rest of the water and to help to avoiding an osmotic shock in the first step of warming. So they are important because both in conjunction, what they do is, as I getting up in concentration, they decrease the freezing point and increase the glass transition temperature. And that made me to go easiest through the ice phase. So it helped me to avoid the ice formation, which is the goal, the final goal in vitrification. But then what cryoprotectants do I choose? So first go to permeating cryoprotectants, and they have a variable permeability, and the permeability changes based on concentration, temperature, meiotic stage, and then they also have variable toxicity. Ethylene glycol is less toxic, but is less permeable. So this is the permeability that has been described with mouse and horses, with propylene glycol, DMSO, ethylene glycol, glycerol. But in cows, they also describe that, for example, at high concentration, 20%, DMSO had similar concentration of ethylene glycol, but at lower concentration, ethylene glycol was better than DMSO. So if we're gonna optimize a protocol, we need to check perhaps permeability of the in the concentration that we are gonna use. What we know definitely is that combination of permeability of permeable CPAs are important, because you can increase survival rate dramatically when you combine cryoprotectants. It has been shown in mouse, in pigs, and in horses. Only mixtures of cryoprotectants have been able to produce blastocysts. Non-permeating cryoprotectants, we have faster equilibration with developmental competence, and triallose had shown some advantage over sucrose, which are the two normal cryoprotectants, non-permeating cryoprotectants that we use, but they displace more water. These saccharides, it means sucrose and triallose display more water than monosaccharides as galactose, for example. In cows and pigs and equine, is not very is very contradicting the information, or is still low information, but it seems that triallose has better fertilization, and at the beginning, Canicin showed that only embryos were from triallose, Meanwhile, in pigs, there were similar rates between sucrose and trehalose. So that's why during my PhD, I tried to investigate different alternatives. So first I went for 3 different non-permeating cryoprotectants, galactose, sucrose, trehalose. And after that, selecting galactose, we went for 3 combination, ethylene glycol DMSO, propylene glycol DMSO, and propylene glycol ethylene glycol. And we found that in equine, propylene glycol ethylene glycol was the best. In cleavage rate was exactly the same that fresh oocytes. And in blastocyst rate, we got a promising 15% of blastocyst rate, but it's still far away from the control in which that replicate that experiment was 40%. But also another important point is that in bovine, we tried 2 different protocols, long protocols, in which the first step was we proved low concentration versus high concentration. And in this point, we demonstrate that if you are doing long protocols, lower concentration is quite better than high concentration. If we move to the cooling rate, this is something that fortunately is quite a consensus about that, because if you have lower volumes, you are increasing your cooling rate, which is super important. Then normally everyone is working with minimal volume device, which it means less than 1 microliter for the vitrification. There are several devices. The most used are OPS, Cryolop, and Cryotop. The open pool straws are every time less used because they keep a bit more of volume, and the Cryotop is the normally most common usage. And the last time for the last factor is viscosity. So probability of vitrification is given by viscosity and cooling and warming rates. So as high these 2 factors, higher probability of vitrification, and lower volume, higher probability. So for this, you can modify the viscosity basically with macromolecules. But the one most used is BSA, and as you can see, as you increase BSA, you are increasing the blastocyst rate. But you have to be careful of your molecule, because if you go 6%, it decreases. You also can combine, but you have to see what is doing with this. Perhaps the pH is going down. And to finish, then the take-home message in the part of the meiotic stage is that there is no consensus about the meiotic stage. It seems to be better efficient than in vitro mature because it has a subtle advantage in gene expression, but it depends on the cumulus cells, are quite specific, and is so important, the fertilization method. Because in a species where you can do ICSI, I think that the best way is not having or having the nucleosides, but if you have to do IVF, you have to conserve as much as possible as you can manage the cell, the oocyte, to vitrify. And in terms of factors of technical factors, we should try to reduce the exposure time and concentration, and then here the mathematical models can give us a hand. Also try to minimize the cooling rate. Obviously, this is with the experience of the embryologists, but we also can try automatic devices that are being tried in this moment in humans. And increase the viscosity, perhaps with substance that doesn't modify that much the pH, or can increase the viscosity with very few amount of the substance, which, like, for example, hydroxypropyl cellulose. So to finish, the main message is that vitrifying oocyte is possible. You can give it a chance. In the last papers about horses and about bovine, you might have between 9 and 15% of blastocyst rate. It's still normally half of your control of fresh oocyte, but we have to continue working. And then with this, I want to finish, and sorry for standing in time. Thank you very much.
Thank you very much, Daniel. It was very, very interesting and informative talk. And I'm sure, again, the audience may have more questions. So, yeah, we'll wait until then so that probably you can answer a couple more questions from the audience. Right. So the last but not least presentation today is from Johanna. Johanna Piibor is from Institute of Veterinary Medicine and Animal Sciences, Estonian Institute of Life Sciences, Estonia. So Johanna graduated from the ENU in veterinary medicine and as a veterinarian, and then she joined with the Combi Vet Eracea research group at ENU as a doctoral researcher. And her main area of research is actually being the use of extracellular vesicles in clinical applications, specifically in reproduction management, as well as in identifying disease etiology in the detritus of the bovine. So today she will give us a talk on unveiling the proteomic landscape of uterine extracellular vesicles in bovine endometritis. Johanna, the floor is yours.
Thank you for the kind introduction, Suranga. My talk is about the uterus. We have heard a lot about embryos, but in preserving fertility, uterus is also a very important part in reproduction. As we know, the uterus is a tight environment that has a crucial role in supporting the development and viability of embryos during pregnancy, especially in cattle, which is my main focus of research, where the implantation occurs around day 19 of the estrous cycle. So during this time, the embryo is solely dependent on the uterine microenvironment and its produced molecules to survive. In this uterine microenvironment, there are a lot of different things that happen. This usually happens and is influenced by cellular communication, which can happen between endometrial cells themselves or between different types of cells. And we know that the cellular communication can do with different molecules. However, the new component that is being researched is this EVs, so-called extracellular vesicles. Extracellular vesicles are lipid bilayer limited nanoparticles that are in the size range of 50 to 1,000 nanometers. These small particles are secreted by cells to different mechanisms, and in general, with these mechanisms, the vesicles are classified into 3: microvesicles, apoptotic bodies, and exosomes. But however, all of these types of nanoparticles contain different cargo, which can be proteins, RNA, DNA, or even metabolites. And when we put these vesicles into transmission electron microscopy and visualize it, we can see these typical cup-shaped like particles. By taking these particles up by different cells, they can transfer this cargo and influence several physiological and pathological processes. So via these vesicles in the uterus, we can influence different processes, either to provide an optimum or suboptimum uterine environment. In my research, I'm more focused on the proteins, one of the part of the extracellular vesicle cargo, which can be in the protein corona around the vesicle, incorporated into the membrane, or even inside the vesicle. As we know, in cattle, the main thing for the farms is to produce new offsprings and get new cows for production. And one of the very fragile periods in cow's life is the peripartum period. Due to this, there is a physical damage to the birth canal during calving, as well as there's sometimes failure to detach the placenta, and also due to the uterine canal being open, there can be an upsurge of microbial contamination in the uterus. However, in general, all these problems should resolve in 13 days postpartum. However, in some cases, these pathogens produce endotoxins or is a cause of the inflammation themselves. And this inflammation of the endometrium can last for a prolonged period, and this can cause infertility. So there are different types of diseases that are classified in the postpartum uterus. And I'm more focused on endometritis, which is an inflammation of an endometrium after day 21. Why I'm interested in bovine endometritis is that it's actually very prevalent in dairy cattle. Around 30 to 70% of cows will have endometritis on the day 21. And some of the cows will be cured themselves, but however, a few of them will have prolonged endometritis, and therefore they're infertile, and this will cause an economic loss for the farms. This disease is generally classified into two in bovine. One is clinical endometritis, which you can see there's this physical appearance of the disease where you can see this mucopurulent pus in the from the vagina. And also there can be subclinical endometritis, which is a very difficult form of disease to diagnose in farm settings because it can only be diagnosed by cytology, where you can see a very increased amount of polymorph nuclear neutrophils. When we look at the during the postpartum period in healthy cows, you have an upsurge of these polymorph nuclear PMMs, and that's totally normal because it's a reaction of the pathogens that will inevitably be in the uterus after calving. But after solving this infection, the inflammation should go down. However, in case of endometritis cows, this inflammation will still stay for a very prolonged period. So I'll tell a bit about my research, which I have done. In my research, I have collected bovine uterine fluid around 36 days postpartum and classified into 3 different categories. One is clinical endometritis, then subclinical endometritis, and healthy. This uterine fluid I have subjected to purification method to get this uterine fluid extra the vesicles. I used differential centrifugation with size exclusion chromatography based method. And I got these small cup-shaped like particles in my uterine fluid. One very simple way to how you actually know how much particles you have in the purified samples is to perform nanoparticle tracking analysis, which is a method where you can measure the size and concentration of the particles. And we already can see that between the different groups, between clinical endometritis, we have between other groups, there is a significantly higher number of particles that can be isolated from this uterine fluid. One is the reason probably for this is that in this clinical endometritis case, we know that their cells will burst. There is a apoptotic process, and during this apoptotic process, there can be released a lot of vesicles as well. While in subclinical endometritis, there is no clinical physical signs. The uterus is apparently healthy, so it really seems like the healthy condition of the uterus. However, there are a lot of differences in the particle size. We can see that in the clinical endometritis, the average size of this extracellular vesicle is smaller compared to subclinical endometritis, as well as in healthy cows. So there is a potential that actually we could maybe use the ratio of the particle concentration and size as one of the clinical diagnostics for identifying subclinical endometritis. But going into the protein cargo of the extracellular vesicles, we also saw a lot of differences between the diseases. We saw that in healthy cows, they're more similar, the cows which chose, and there is a differentially clustered between the clinical endometritis and subclinical endometritis. And if we look at the heat map, we can see that these different types of diseases have different protein profiles. We identified in total 1,549 proteins that were significantly different between. We saw identified 1,549 proteins, and from this 248 proteins were significantly different between the groups. And with this, we saw that in clinical endometritis and healthy cows, the first cluster are clustered similarly with similarities in protein expression of cell apoptosis, cell migration, and immune-related processes, while healthy cows and subclinical endometritis have enriched proteins in the functions of regulation of translation and gene expression, cellular nitrogen compound metabolic process, plasma membrane organization, and regulation of cell shape. Then we subjected proteins to gene ontology analysis to see these immune-related pathways, because veterinarians have not been still sure if clinical endometritis and subclinical endometritis are the same diseases, just in different forms, or they're actually two separate diseases. And to figure this out, we compared healthy cows with clinical endometritis as well as healthy cows with subclinical endometritis. And we saw that only 15 immune-related pathways were considered consistently identified in both the subclinical endometritis and clinical endometritis, while in clinical endometritis, there were 4 unique pathways, and in subclinical endometritis, 20 unique pathways compared to the healthy cows. This would indicate that there is potentially a different inflammatory response between the different diseases, might indicating that the causes of this disease might be totally different. And when we've compared subclinical and clinical endometritis, we confirmed that there are differences between the diseases, and potentially there are different mechanisms. Traditionally, veterinarians believe that during peripatrician period, inflammation development involves recruitment of leukocytes, which helps to eliminate the infectious agents before undergoing programmed cell death. And with this failure of apoptosis and the incapability to counteract bacterial harm, there is, and the persistent activation of pro-inflammatory pathways may play a role in chronic inflammation. We can see that there are different responses in apoptosis between the disease, where in clinical we can see the activation of the apoptose response, which does not occur in subclinical endometritis. However, there is an activation of different mechanisms, for example, activation of cell morphogenesis, leukocyte profilation, and regulation of cell shape in subclinical endometritis compared to clinical. Therefore, there are potentially different disease mechanisms. In summary, we saw that clinical endometritis and subclinical endometritis have totally different disease responses, with particle concentrations significantly higher between healthy cows and clinical endometritis, however, not between healthy and subclinical disease. The protein profile between the groups were significantly different, as well as there's distinct expression patterns indicating varying immune response and separate disease mechanisms in clinical endometritis and subclinical endometritis. The reason why we should research disease is that it causes a lot of infertility problems, especially in dairy cattle. As a subclinical endometritis is very difficult to diagnose in farm settings, we potentially need biomarkers for the quick diagnosis of the disease, and then therefore we can develop different therapeutic tools to treat these cows. Also, we have a lot of questions about the etiology of disease. Is subclinical endometritis caused by similarly as clinical endometritis? As we know that in clinical endometritis, it has been confirmed that there is a bacterial cause of this disease, and treating with antibiotics actually helps to treat this disease. However, it's not same in the subclinical form. Treating with antibiotics does not help to increase the fertility in cattle. But if we would know the reason of the cause of the subclinical endometritis, we can develop prevention of tools for endometritis. Thank you. And I'm open for questions. I guess we're all open for questions now.
Thank you very much, Johanna, for sharing your interesting data. So I think it's the time for questions and queries or clarifications. Please, if you want to ask from yourself, please unmute yourself and ask the question. And also we are open to the chat option as well. Yeah, please, Claudia, please go ahead.
Andres, I was actually wondering whether you had tried the IVF technique that Katrine Hinrichs had published. Whether it's also, do you know, is it working in other people's hand?
Hi, Claudia. I haven't tried it myself, but I know that the group in Belgium tried it and they got a fall out of it. So I know that some people is trying that same protocol. For some of them it's working, for some of them it's not. So I guess it's still a little bit tricky. But I know it has been possible again. Maybe Danielle, you can comment on you're there, so you probably know more about that than me.
Yes, we had a fall with the IVF protocol. Now there is a PhD student actually working on that, and I'm going to vitrify now an embryo from IVF, so it works. In our hands, we don't have the same success that Katrine Hinrich said. So in our hands, we have been trying perhaps 4 or 5 times, in which the fall was quite fortunately because it cleaved one oocyte cleaved from 60, and that one make an embryo, and that one make a ball, and perfect, everything. Now this last time, like 7 oocyte cleaved out of 20, so it's lower than the ICSI normally. And from that 20, we got one embryo that I'm gonna vitrify. So it works, but the protocol, you have to be very careful in several steps. And it's something that we are gonna work in, but I think that the stallion is really important. You have to start with an amazing sperm, I think.
Yeah, they also recently showed us some data on the ICER in Brazil this year, because all these protocols were performed with fresh semen, and they have now modified the protocol to be able to produce blastocysts with frozen semen. So apparently in their hands, IVF, it's also working now with frozen semen, but I agree that we still need to make this, more repeatable across different laboratories. It's not yet there.
Thank you, Andres. So we have one question in the chat. I think it's directed to Daniel. Does vitrification affect the success of sperm to fertilize the oocyte?
Sorry, the vitrification of sperm, or?
I think Luciano is here. Luciano, can you elaborate a bit more on the question that you want to ask? I think he's wondering about the sperm functionality.
Yeah, I guess I can read his mind since anyways, we work together. We've been wondering about Daniel, what we had briefly talked about, the zona pellucida hardening. I guess because in equine you do ICSI, so you don't have to worry about it, but in bovine you potentially have to worry about it when you do IVF following vitrification.
Yes, that's something that it will be nice to do research about. I mean, it is well known that vitrification, one of the effects on the oocyte is the hardening of the zona pellucida, but it's very little research about how much is the hardening. So because for example, I recently received a paper in which in pigs with that the polyspermic is a big problem. They did a protocol to try to make harder the zona pellucida, and it's also a problem in bovine. We are doing some research in polyspermy, and we have seen some bulls that have up to 50% of polyspermy. So I think that vitrification do make harder the zona, but the matter that it affects the fertilization or not is gonna depend on the concentration what you do IVF and in the bovine case, the bulls that you use, because they are bulls that has very high polyspermy rate and very good penetration rate. So in that bulls, I don't think it affects, but the other bulls that have lower penetration rate, it might be affected by the zona hardening.
Any more questions from the audience? Yeah, maybe I have a couple of questions actually. Actually, I would like to ask the first question from Claudia, just a clarification. It was very fascinating to see the single cell sequencing data integration that you have shown. So what we experience in our case, usually in the case of proteins, usually when you go by bovine and human, if you try to figure out even one single protein which is common, it is very, very difficult. So in your single cell data set, what were the kind of markers that you use to classify these cells? I mean, it's really matching. I was really fascinated to see that UMAP plots that you were showing.
I guess it really, and so the tool really only works when stuff is similar, because at some point, just to sort of test the robustness of the algorithm, we were taking our day 17 trophoblast cells and we're trying to align them, I think, to second or third trimester pregnancy trophoblast. And there the algorithm says, sorry, I can't integrate because there's not enough similarities. So I guess because at this point with the single cell, I mean, you capture literally, it's such a sensitive technique, you can capture down to something like, 15, 16,000 transcripts. There are enough similarities between the human cells and the bovine cells. And sort of the internal control is also, the human data set also included earlier embryos that still had sort of the classic trophectoderm aligns with the interferon tau, sort of our classic trophectoderm cells, and then our specialized trophectoderm cells in the bovine, like the binucleate or this unknown population, align more with the specialized cells, like the cytotrophoblasts and so forth. So I'm still amazed how these algorithms work, and again, how robust they are, because I think it's a good control. Like the second or third trimester trophectoderm cells, the algorithm gave us an error message, said, well, not really an error message, the cell populations were separated. It wasn't able to match it. So yeah, it just, because you're doing it at the whole, virtually at 15,000 transcripts, there's enough similarities between the different cell types.
Thank you. I think Daniel has raised the hand first before Andres, so I'll give the chance to Daniel first. Yeah.
Yes, thank you. I have a couple of questions for each presenter.
Please go ahead. Yeah.
So I will start with Andres, which I want first a curiosity. When you inject with the koala, you also did with koala oocytes? And did you got embryos?
We were trying at the beginning. So what we are doing is we are injecting koala sperm into bovine oocytes. That's the first experiments that we did. We call that koala embryos, because we wanted to know if the koala sperm was able, there's a bunch of new stuff for me since I'm in Australia about marsupials, right? I don't want to spend too much time in marsupial reproduction in this seminar, but it's fascinating how these guys and the sperm physiology of these guys, because they can last like 40 days refrigerated, the semen, but they are really, like, if you try to freeze semen from koala, it's really bad. But because no one was, attempting or doing ICSI with koala, I said, well, we can totally do ICSI. So a few sperm were moving, and we are looking at pronuclear formation after injection of koala sperm into a mature bovine oocyte. And we are seeing that actually a koala sperm can induce pronuclear formation and induce oocyte activation in cattle. And we had the chance to inject koala sperm into koala oocytes as well, just once. But oocytes, like this is not known. Like, we don't know oocyte maturation in koala. We don't know the media. We don't know anything. So I just replicated whatever we're doing in horses and cows. I got like a few oocytes with polar body, but none of the oocytes went anywhere. And we are collecting samples from dead animals. So this was a koala female that was in a wildlife hospital. She was, suffering a it was yeah. The idea, we are setting up grants to try to set up these technologies in marsupials. The next step will be injecting these koala sperm into kangaroo oocytes, because that is, a little bit more close than the cow. And we have way more access to kangaroo oocytes because kangaroos in Australia are overpopulated, and we can, go and collect ovaries pretty much right away after the kangaroos are being shot. And that's better. So we are working on kangaroo oocyte maturation now. and probably soon gonna have some more data to share.
Nice. Continue with Dr. Claudia. You show amazingly that the bovine is might be a good, perfect model for the human in the embryo. But then have you do the similar analysis into the bovine transcriptome with bovine or in any other animal comparing with humans?
You mean like mouse or something?
Well, I mean, if you showed amazing transcriptome and comparative bovine blastocyst against human blastocyst, have you done the same with oocytes transcriptome single?
No, the oocytes are too big for the system, sadly. Because I was really excited when I first learned about 10x genomics technology. But the problem is the microfluidics device has a limited size. And so the oocyte was over 100 micrometers. The cell type is sadly too large for this system. So you can't analyze oocytes using this technology because it's the cell is too big.
Okay. Thank you. And the last question to Joanne is, have you evaluated the microRNA content of the EVs to try to look for biomarkers of endometritis or something like that?
No, we haven't done that. Bovine endometritis and extracellular vesicles is a very new field. There is literally no papers before published about vesicles in endometritis cases. So we first figured that we will focus on proteins.
Andres, you had a question. Yeah.
Yes, I do. Thank you.
Go ahead.
Claudia, you show us at some point. You show us the data. First of all, it's amazing how I never thought that it could be that similar, human to bovine. Like I wouldn't expect that at all. Which is super interesting. I have two questions. One is at some point you show us the data of the single cell sequencing on the embryonic disc of the 3 embryos, and 2 embryos that look really similar, but in one embryo you have a missing cluster of cells.
Endoderm.
And I was like, why is that missing? Do you have any comment on that?
The endoderm was missing.
Okay.
So it was the youngest embryo, and there the endoderm was missing. We had really surprisingly little endoderm cells overall, and that was the youngest embryo. So either it didn't have any endoderm yet. That was also the embryo that only had immature mesoderm. So it was and so either the embryo, either we missed simply the cells with the digestion process, or it didn't have any endoderm cells yet.
So, but are you sure that that's a 17-day embryo, right? Because you know when the cattle ovulated and inseminated. So it's not a late embryo. Well, you never know. All right. Nice.
Yeah,
very nice.
I guess it could be a day 6. As I said, we were not doing any synchronization because we wanted sort of like a natural cycle. Mm-hmm. And so, we didn't really track the ovulation because in Germany, the animal care rules are very strict. And for example, I forgot to apply that we could do ultrasound to track ovulation. And then I'm not allowed to do that. So we were simply basing it on visual heat observation. And they also have the SensUp, the automated heat detection system. And so we based the time of ovulation based on that observation. And then based on that, we were determining day 17.
Yeah, and the second question is regarding like your maybe future experiments, trying to understand the impact or helping to understand more assisted reproductive technology impact on embryo quality. And are you planning to use like this outgrowth of trophoectoderm cells to perform RNA-seq? Because in my what I've seen, it's very easy to do with horse blastocyst. So you can just it's not that hard to perform trophoectoderm outgrowths on blastocysts with in vitro produced blastocysts in horse. So I think it could be like a nice model, and you're not dealing with animals too much, you know. It's your plan to make everything on day 17 with IVF and.
No, I'd be if you're interested on working on this together. I don't have any access to horses here, but sadly, well, maybe I can hide some horses somewhere. That's what I keep telling the people in the barn. But if you're interested on working on this together, and like if you generate the outgrowth, we could try to analyze them. The challenge is, I guess, they're constantly developing their technique, but the best data you get when you have fresh digested cells. But I mean, one could also consider, for example, to cryopreserve the cells, for example, for shipment, then thaw them, wash them, and immediately process them through the device, for example.
Yeah, that's something that can potentially be done. Awesome, thank you.
Yeah. May I ask a question from Daniel, because it was fascinating to see this, again, the cryopreservation of oocytes. So I was really interested some time back on cortical strip, vitrification. I have been working a little bit on that. So did you ever try to use the cortical strips, the whole strip, and then did you compare the success rate between the whole tissue versus individual? Individually or group-wise, vitrified oocytes.
Sorry, compare what? The cortical
strips, from the slaughterhouse samples. I know Teresa's group, from Northwestern, they were really doing a lot of work on vitrifying the whole, the cortical strips from the ovaries, from the slaughterhouse samples, and they were claiming that it's better when you vitrify as a whole tissue rather than you take it out and do maturate and then vitrify. I mean, recovery rates, everything were higher. I think they published one paper sometimes back as well. Have you ever had any such experience, especially in the case of equine in horses? The vitrifying the whole strip, they slice it like very tiny.
But this will be perhaps like another technique, which is cryopreservation of ovarian tissue.
Yeah.
I mean, it's a possibility. We haven't tried in equine. We have some experience with feline and dog oocytes, in which we didn't do development of these follicles. We just make histopathology, and we determine like a kind of protocol for that. But the problem of the preservation is that of that tissue, that it depends what you are gonna to use it. In humans, it's a successful technique also now to transplant this tissue after vitrification again into women, and they have been able to recover folliculogenesis. We haven't tried that in animals. The problem that they have experienced in humans is that if they do it in women with cancer, they have got some cases in which the cancer is again transferred. So this is the limitation. And the other limitation, if you do it in vitro and you just want to grow the follicle in vitro, then you have to do all the technique of growing follicles in vitro, which is quite tricky also. But there are quite two different techniques: so ovarian cryopreservation and oocyte cryopreservation.
Okay, thank you. And my last question would be targeting Andres. So I was really interested to see this lyophilized sperm thing. So how was the success rates when you compare with the fresh and lyophilized? Have you had any experience in the case of ICSI?
I haven't. We are doing freeze-dry koala sperm at the moment, and the success rates in general are low, very low compared to other preservation methods of sperm. And in the case of the horse, there was a report by the group of, again, Catherine Hinrich and Choi in 2011, and it was the only one that was produced, because it's pretty much when you freeze dry sperm, you kill all the sperm. And what you are trying to preserve really is the DNA. And the problem is that you're also messing up with some proteins that are in charge of, triggering oocyte activation. So it's improving. There are a few new protocols that are working better. But I mean, I think this is a little bit of the future of the thing, like how I think it's gonna move the industry. Because, for example, in mice, they've been able to freeze dry sperm. They send that sperm to space. They heated that space. They heated the sperm to 150 degrees. You can do anything to the sperm, and then you will have live, healthy offspring out of that. So it's an amazing way of preserving cells or DNA, I would say. And I think that definitely it could change the way that we are thinking on, biobanking or simplifying sending samples across places. And it's the potential is very big, but the efficiency is still very low. There's still very low because there's no way of really selecting the sperm after the process, because the sperm is not moving. So you cannot select the sperm. You cannot use, new devices or microfluids or any of the new technologies that we have now to select sperm. It's just pretty much your eye. But yeah, there are new protocols that are preserving better the DNA. And actually in birds, they got a protocol, they set up a protocol where they got 3% or 1% motility after freeze-dried sperm. And if you think about it, that's pretty amazing, because even 1% motility for ICSI means that you have a motile sperm. So that's a lot. Yeah.
Thank you. I think Daniel has one more question.
Yeah, just two very technical questions for Andres. The first one was more or less answered at the end. So how do you select the sperm when you work with dry freeze sperm, if you don't have motility?
You don't. You just select whatever sperm is. The thing is that after after diluting, because you just add water to the powder sperm, right? And you put that in the solution that you're gonna do ICSI, all the sperm is dead. A lot of detached head, tails, and so it's a little bit of a mess. So what you try to find is sperm that looks intact, or as alive as possible, but they are all dead. Really, the membrane is broken in all of them. But that's not the issue for ICSI, right? You want to break the membrane of the sperm. The thing is that there's currently no way, as far as I'm aware, of assessing DNA quality without killing the sperm or without affecting sperm viability. So I think that if we at some point can develop a technology that we can look at DNA quality before injecting the sperm, that can be, you know, like a very useful tool to select sperm before injection. Not just for freeze-dried sperm, but for any other ICSI. But I'm not aware of that technology yet. Probably we're going to have it at some point.
Good. And the second question is this even more technical, but I just realized that when you took the sperm from the koala, you took it from the head. And normally when you're going to inject, or when I'm injecting, I normally try to put the head in contrary. So do you normally inject in any position, or do you think it's no difference? Or
yeah, no, so like, look, that video, it's a very funny video because that was the first time I ever took a straw out of a liquid nitrogen tank. So I didn't even know if the sperm was, you know, big, like the big that will not get into the human ICSI needle. I wasn't even know how the shape of the sperm was. So I was literally playing with the sperm. I was trying to kill the sperm, and the head of the koala sperm, now I'm becoming an expert on this, but it's like an S, but I don't know if you saw in the video. But the problem is that sometimes, I mean, I can grab them from the tail, but the head will get stuck because of the shape on the tip of the needle. But most of the ICSI that I've done with bovine oocytes, I did it like from grabbing the sperm from the tail, so, you know, directly. But I actually have a master student that did it, like a very small experiment, injecting from the tail or the head. And there's no much different, like if you're successfully on, you know, breaking the membrane of the oocyte, you can inject. And for the students that they are not very familiar with ICSI, I will say that one of the most tricky part for them is to grab the sperm from the tail. It's very easy. It's more easier for them to grab them from the head, right? So if you have the system, the setup, the oil or the air or whatever you have to micromanipulate, if everything is working perfectly, just a very few times grabbing the sperm from the head will mess up, you know, the sperm moving up and down. Usually it's just fine. I think you can inject it both ways, but I guess that it makes more sense to just inject it from head first, right? But yeah.
Okay, with the interest of time, I think we should. Oh, there's one last question from.
I just had one comment. I remember I read a publication at some point where they retrieved a sperm from like a formalin fixed section in mice and did ICSI and got a blastocyst. So I guess sperm are very, very hard takers. So
Let's take it like
yeah,
that.
if you think about it, how the sperm is, the DNA of the sperm is packaged in some like a very particular way. So it's a very, very hard thing to mess up. So and it's millions of them. So it's really fascinating, and with the use of technology we can, yeah. So Daniela, I have like a quick question. If I can ask. I will start doing some experiment on vitrification of marsupial oocytes at some stage, because that's what is abundant in Australia. And they have like a very thin zona pellucida, but they are big. They are like 150 microns, and but they look pretty much like a mouse. So, you know, they are like clear. They don't have like these lipids that we have like in horse or bovine or pigs. Do you think that, like, how important will be the size of the oocyte to adjust, cryoprotectants? Is any way of, you know, formula to, if you have like a bigger oocyte, will you increase any particular cryoprotectant to make the process more efficient?
No, I don't think it's. I mean, it's important, that part of volume, size, and everything. But you are talking about 150. So with the zona, even if the zona is thinner, like the old lemma might be then 120 or something like that. So I don't think you have to adjust too much the protocol. You can try just with the ones that we use in humans or equine. What can help you a lot is to denude an oocyte and put it with the micro manipulator in a droplet of cryoprotectants, and then see how fast it shrunk and recover. And then it will give you a excellent idea of how is the permeability and how, if it's similar to the models that they have published in all the species, basically, you don't have to adjust anything. If it's lower, then perhaps you have to increase times or whatever.
That's interesting. It's just placing an oocyte in the first step one of commercial.
Yeah, more or less in the equilibration solution. So what I do is take one droplet of holding and next to it one droplet with the solution, and I grab the oocyte with 2 holding pipettes, and I just move it from one droplet to another. Well, just to see it and can measure and be able to measure in the microscope. Because if you just move it with your pipette, the oocyte is gonna float, and then you have a estimation on how it shrinks, but you cannot film it to see in seconds, because normally it's the first 20 to 30 seconds that it's gonna shrink to the minimal volume.
And is like, so you move the oocyte from the holding to the oil, and then from the oil to the other droplet?
No, not to the oil. You have a ICSI dish in which you have the droplet as close as possible to the equilibration solution, and you just move it through the oil to the
but I mean, like,
droplet.
the whole oocyte has to go through the oil from move one solution to the other. Right?
The other possibility is that I don't have it here, but the other possibility that I have seen in paper is that they hold the oocyte with a holding pipette, and in the other injector they put a needle that is big enough to hold the all oocytes, and then you move through the oil, protecting it with this big pipette. And then you remove it.
You don't really need the oil, right? Like, why you need the oil? Like, we can just do it with droplets without oil. The process is pretty fast.
Yeah, perhaps. Yeah, I haven't tried without oil. I don't know.
Okay.
If you move it in the holding pipette from one droplet to another without the oil, perhaps it works also. Yeah. I have tried only with oil. I don't know.
Okay, awesome. Thank you, Danny.
Okay. With the interest of time, then let's wind up the session. So I would like to thank all the speakers. Of course, Claudia is part of this Baltic grant, which we got a small grant program. So on behalf of myself and Claudia, and even EMU and from the Federal Institute for Animal Research, on behalf of both the institutes and both the research groups, I would like to thank Daniel and Andres especially for joining us today. And I hope this would open up some avenues to do some future collaborations. And it's really great to hook up with Andres after like 10 years. So science is an amazing place where you will meet and greet people time to time. So thank you very much, and have a very good evening, and also have a very good day to Andres, because I know it's morning. And thank you very much for joining us. It was really, really amazing talks that we had.
Thank you for the invitation. Bye, everyone.
Bye. Thank you.